Good morning, everyone. Welcome to Zoology online Guru classes. Today we are going to discuss about the sexual reproduction of flowering plants. That is important for NEET examination as well as MCERT and IP examination also. Before going to the chapter sexual reproduction of flowering plants, first we should understand the life cycle of flowering plant. Life cycle of flowering plant. So here in the NEET examination, we may get 3 to 4 questions. In MCERT examination, we'll get 2 questions. IP examination, there is a long answer, right? So here, let us start with the sporophytic plant. So here, sporophytic plant means which contains true roots, true stem, true leaf, true flowers. So here in the image, it is seedling. So from the seedling, that is nothing but a plantlet. From the plantlet, a complete plant will be formed that contains true root, true stem, true leaves. So here, so flower is the reproductive organ. Here, this is the structure of flower. It has mainly two important organs that is important for reproduction, that is androecium and gynoecium. The unit of the androecium is the stamen. The unit of the androecium is the stamen, and that is usually represented with the letter A, and the unit of the gynoecium is a carpel. It is represented with the letter G. So this carpel is also called as pistil. This carpel is also called as pistil. So first let us talk about the androecium, that is stamen. So stamen has mainly two parts, that is one is a filament and another one is a anther. So this one is a filament and this one is a anther. So together filament and anther is known as the stamen. In detail we are going to discuss in the chapter about the structure of stamen. So here this anther contains the sporogenous tissue. In the sporogenous tissue, pollen mother cells are present. So those pollen mother cells undergo PMC, we can call it as PMC, or microspore mother cells. Microspores mother cell, so which is diploid in condition, right? So this PMC undergoes meiosis division, also called as reductional division, So this PMC undergoes meiosis division to produce the haploid microspores. These haploid microspores all are they are associated together with the help of a substance known as callose. So callose makes these microspores attach together, right? And in the microsporogenous tissue, tapetum is present. So that tapetum produces the enzyme callase. This callase break down the callose. This callase enzyme break down the callose so that all the microspores dissociate with each other, and now the microspores all are free microspores. These microspores also called as the pollen grains, and these pollen grains are the young and unmatured pollen grains. Right, all are haploid in nature. So this dissociation is due to the enzyme callase as well as due to the dehydration, and as well as enzyme callase is involved in the dissociation of this microspores by breaking the callose, and so that these tetrads will now the microspores free. These are called as free microspores or pollen grains, and each microspore, here all the microspores are functional, all are potential. All the microspores are functional or all are potential. Means every microspore can give rise to a male gametophyte. Every microspore can give rise to a male gametophyte which carries a male gametes, right, which is involved, which is important for the fertilization process, right. So here this each microspore, young microspore or unmatured pollen grain, it undergoes mitosis 1, mitosis 1 division to produce the vegetative cell and generative cell. So this vegetative cell is usually bigger and generative cell is smaller. This vegetative cell contains the abundant food materials and generative cell is involved in the production of 2 male gametes by undergoing the mitosis division. So here this pollen grain, nothing but it is a matured pollen grain. Now it is known as a matured pollen grain. We are going to talk in detail about the pollen grain structure in the pre-fertilization events, that is after completion of this life cycle, right. So here how many pollen grains are formed? 4 pollen grains are formed and all are active, all are functional. When this pollen grain land on the stigma, that is a gynoecium unit is the stigma, and here this carpel or pistil contains mainly 3 parts, that is a ovary, style, stigma. When this pollen grain land on the stigma, if the stigma accepts the pollen grain starts germination and it produces a pollen tube, and that pollen tube enter into the ovule through the style and it reaches the mature egg embryo sac. So there in the pollen tube it carries the 2 male gametes. These male gametes are formed as a result of the mitosis 2 of generative cell. So this generative cell undergoes mitosis division to produce the 2 male gametes. So these male gametes are finally reach the mature egg embryo sac, thereby fertilization takes place. So how the fertilization takes place, all these events we are going to discuss in the chapter, right. So due to the fertilization, means egg cell fuses with this male gamete, and due to the fertilization a zygote will be formed, which is diploid in nature. A zygote is diploid in nature, and from the zygote embryo is formed. From the embryo a small plantlet is formed. From the plantlet complete plant is formed, right. This cycle alternatively repeated, means first initially sporophytic generation. Sporophytic generation is a dominant stage of plants, that is from zygote to pollen mother cell considered as a sporophytic generation, and gametophytic generation is nothing but formation of gametes from the pollen mother cell. And here how this megaspore is formed. Now let us talk about the gynoecium megaspore mother cell. So we had talked about the pollen mother cell and how the pollen grains are formed. Now let us talk about the how this mature egg embryo sac is developed. So just like androecium, in the microsporangium is present in the anther that possesses the pollen mother cell, and here in the gynoecium part a megasporangium is present. That megasporangium contains the megaspore mother cell, which is diploid in nature. So this megaspore mother cell can able to produce. It undergoes meiosis division to produce the linear tetrad megaspore, and out of these 4 megaspores, 3 cells will be disintegrated. Only one is a functional megaspore. It is completely contrasting to the pollen grains, contrasting to the pollen mother cell. Pollen mother cell produces the 4 pollen grains, and all the pollen grains are functional in nature. But here in the megaspore mother cell, it produces the 4 megaspores. Out of the 4 megaspores, 3 cells will be degenerated. Only one is the functional megaspore. This functional megaspore finally developed into mature egg embryo sac, or it is also called as a female gametophyte. When the pollen tube reaches the mature egg embryo sac, there fertilization takes place and it results in the formation of zygote. That zygote undergoes different stages to form embryo. From embryo, a plantlet is formed. From the plantlet, a complete plant is developed, and that is known as a sporophytic plant, which contains true root, true stem, true leaves. Okay. So the cycle alternatively occurs. Initially, sporophytic generation and gametophytic generation. Gametophytic means formation of gametes, that is male gametes and female gametes. Okay, right. Now let us talk about the pre-fertilization structures and events. So in this chapter, this is a important topic. From this area, we may get one question in the examination. So initially, several hormonal and structural changes takes place, and that results in the formation of floral primordium. Inflorescence bear the floral buds and then flowers. So in the pre-fertilization structures and events, first let us talk about the androecium. Androecium is the one of the important part of flower that usually represents the male flower. Here it is a male representative of the flower, and here it consists of filament and anther. Together, filament and anther known as the stamen. The proximal end of the filament, this is the proximal end of the filament, and it is the previous NEET examination question. Proximal end of the filament attaches to which part? It attaches to the thalamus. It attaches to the thalamus or petals. And the distal end, this is known as the distal end. The distal end of the filament attaches to the base of the anther, usually called as connective. In the NEET examination, proximal end of the filament attaches to which part? Asked. That is attached to the thalamus or petals. Okay, right. So here the number of stamens vary from plant to plant, right? And then usually in most of the flowering plants, these anthers are dithecous anthers. So when we make a horizontal cut of the anther, that is usually called as transverse section. It is bilobed, and each lobe contains 2 theca, and overall it is known as the tetrasporangiate structure. We shall discuss about the internal part of the TS of anther, transverse section of anther, and it mainly contains 4 parts. And remember, most of the flowering plants are the dithecous anthers. Some exceptions are there. The exception is the hibiscus. This hibiscus is also called as a China rose. In the hibiscus, we will find monothecous, and this hibiscus belongs to the Malvaceae family. Right? In most of the flowering plants, anthers are dithecous, but exception is the hibiscus rosa sinensis. usually called as China rose, and the anthers are monothecous, and it belongs to Malvaceae. Now let us discuss about the uh, microsporangium wall layers of this transverse section of anther. Okay. So this is the structure of microsporangium, and it is a transverse section of anther. So here this is the right right anther and filament. So when we cut the anther, so it is visible like this tetras. It is a near circular in outline. Very important. This microsporangium is near circular in outline. So focus on this word in the examination. So examiner may ask which of the following statement is right. In that, to confuse the students, he may ask just the microsporangium structure is circular in outline, but it is not circular in outline, it is near circular in outline, right? So when we cut the anther, when we observe under the microscope, this transverse section is looks like this. So here it is a bilobed anther. So this is a lobe, and it is another lobe. In each lobe, two theca are present. In each lobe, two theca are present. That is why it is called as a dithecous anther, and the structure is known as the tetrasporangiate structure. Tetrasporangiate, because here in the microsporangium structure, that is it is one microsporangia, it is another sporangia, second, third, fourth, all are located at the corners. In each microsporangia, number of pollen mother cells are present, or microspore mother cells are present. So here in the center, when the anther is young, it is in the center a compactly arranged homogeneous cells are present, and that is called as the sporogenous tissue, right? So here it mainly contains four wall layers, that is epidermis, endothecium, and middle wall layers. So these are the middle wall layers. Usually they are 1 to 3 layers, and last one is the tapetum. These are the four important parts of the microsporangium, right? So here there is another structure, that is the connective. This connective is mainly composed of some tissues called as vascular tissues. When the filament attaches to the anther, so this anther is projected into the anther, and that attaches to the vascular tissue, and that is known as the connective. Okay, right. So here mainly it has four parts. When we observe the this microsporangium under the electron microscope, and here some part of the microsporangium is enlarged here, so that we can clearly label the things. First, the outer layer is called as the epidermis, and below the epidermis endothecium, and below the endothecium middle wall layers, and the innermost layer is the tapetum. So in the tapetum microspores mother cells are present, right? So first let us talk about the epidermis. So epidermis is generally one celled thick. It is generally one celled thick, right? Here in the epidermis, we need to remember one important point, that is stomium. Stomium is nothing but a specialized cells are present in the grooves of the anther, especially in the epidermis region, in the epidermis region. Some grooves are present. At the grooves, stomium is present. It helps in the protection and dehiscence of anthers, right? So the main function of the epidermis is the protection and dehiscence of pollen sacs or anthers when they become mature, right? So here the formation of microspores takes place from the pollen mother cells, pollen mother cells, right? So here this is the epidermis. In the epidermis, you have to remember just 2 points. One is it is only one cell thick, and some specialized cells are present called as the stomium, and function of the epidermis. That is, it helps in the and dehiscence of pollen sacs, and endothecium. This endothecium is a second wall layer. Here the cells expands radially. Radial expansion of cells takes place, and it contains mainly fibrous thickenings at the corners. Fibrous thickenings at the corners of the cells present in the endothecium. At maturity, the cells they lost the water. When the cells of the endothecium become mature, they lost the water and become contractile. And the function of the endothecium is a similar function to the epidermis, that is protection and dehiscence of pollen sacs or anthers. Come to the middle wall layers. So these middle wall layers are usually 1 to 3 layers. This is usually 1 to 3 layers, and the function is just like epidermis and endothecium. Same function, that is protection and dehiscence of pollen sac, right? So these microspores mothers, this middle wall layers degenerate after some time when the anther become mature, and the most important is the tapetum. So tapetum is the innermost wall layer. It is also known as the nutritive tissue. So tapetum is also known as the nutritive tissue. So this tapetum nourishes the developing pollen grain. Means for the developing pollen grains, food is required. That food is supplied by this tapetum. So abundant food reserves are present in the tapetum. In the tapetum, cells are usually larger cells, compactly arranged, and dense cytoplasm, and we can observe the more than one nucleus. So cells are larger, and dense cytoplasm is present. More than one nucleus, that is sometimes binucleate or multinucleate condition is observed, right? Here pollen mother cells are present. So each pollen mother cell undergo meiosis division to produce the 4 microspores known as the pollen grains, right? So this is about the wall layers of the microsporangium. You need to remember all the 4 parts: epidermis, endothecium, middle layers, and tapetum. Tapetum is important. This regarding the tapetum, previously asked so many times regarding the tapetum. What is tapetum and what is the importance of tapetum? So tapetum is the innermost layer, and it is also called as a nutritive tissue layer, right? Cells are larger, compactly arranged, dense cytoplasm is present, and more than one nucleus is present. So this is about the structure of microsporangium, and here this structure is called as a tetrasporangiate structure. So in most of the flowering plants, anthers are dithecous, and dithecous anthers have this type of tetrasporangiate structure. In Malvaceae, that is in hibiscus, that is only single lobe is present, so that is known as a bisporangiate structure. In hibiscus, right? Now let us talk about the microspores means pollen grains. So the structure of pollen grain and importance of pollen grains. Microsporogenesis. So microsporogenesis is nothing but a formation of microspores from microspore mother cell. As the anther develops, each sporogenous cells undergoes meiotic division, that is a reduction division, to produce a microspores. So all the microspores are functional in nature. Each microspore can gives rise to a male gametophyte which carries a male gamete. So this is a microspore. This microspore undergoes mitosis one division to produce a two cells, that is one vegetative cell and another one is a generative cell. This is known as a two-celled stage. Most of the flowering plants, the pollen grains are shed at this two-celled stage. That is, in 60% of the flowering plants, these pollen grains directly land on the stigma and then initiates the pollination and then fertilization. And remaining 40% of flowering plants, that is angiosperms, they shed at three-celled stage. That is, one vegetative cell and the generative cell undergo mitosis division, produces the two male gametes. After the production of two male gametes, the generative cell degenerates itself, and this is known as three-celled stage. So in the 40% of flowering plants, pollen grains are shed at this stage. That means this pollen grain land on the stigma, initiates the pollination and fertilization. So this question is asked previously in the NEET examination. That is, in 60% of flowering plants, pollen grains are shed at which stage? That is two-celled stage. In remaining 40%, shed at three-celled stage. Here also this generative cell undergo mitosis division, but when it occur means when the pollen grain land on the stigma, thereby germination of pollen tube. During the germination of pollen tube, generative cell undergoes mitosis division to produce the two male gametes. Here in this case, directly generative cell is produced in the mature pollen grain before landing on the stigma. It is after landing on the stigma, after germination of pollen tube, two male gametes are produced as a result of mitosis 2. This is about the microsporogenesis. So microsporogenesis means formation of microspores from the microspore mother cell. This microspore mother cell is also called as a pollen mother cell. Microspores are also called as pollen grains. Next, we are going to discuss about the structure of pollen grain in detail, and some important and interesting facts are there. So very interesting. Thank you for watching. Please subscribe and share. Thank you.